You're listening to Conferences Online Allergy from Children's Mercy Hospital in Kansas City, Missouri. Today is October 14th, 2024. Our topic is Abbas Chapter 2, Cells and Tissues of the Immune System. This is presented by Dr. Nikita Raje. She's an Associate Professor of Internal Medicine, Pediatrics, and is the Chief in the Section of Allergy Immunology at Children's Mercy Hospital in Kansas City, Missouri.
Today is October 14, 2024. We're going to be talking about Chapter 2, Cells and Tissues of the Immune System, and from Abbas, and I'm Nikita Raje from Children's Mercy, Kansas City, University of Missouri, Kansas City. Let's get started. Hope you guys got a chance to read this chapter. My disclosures are mentioned here, nothing relevant to the talk today. The learning objectives are to recognize the structure and then the function of various immune cells, and then we'll identify the differences in the structure of various lymphoid organs. Here's the outline. We'll talk about the different cells and then the lymphoid tissues. A couple of pre-test questions. We'll come back to these at the end. A drug that blocks the function of chemokine receptor CCR7 would result in which of the following abnormalities? And then in 22q11.2 deletion syndrome, the thymus fails to develop. Which of the following characterizes the immunodeficiency state in this syndrome? All right, so we'll come back to those. We know that the cells of the immune system are white cells coming from bone marrow, and there are different types of white cells, and just mentioning the various normal ranges for these cells. So neutrophils being the most prominent, then lymphocytes, and then the other cells, eosinophils, basophils, and monocytes. So let's talk about each of these. So going back to medical school, really, talking about neutrophils, these are also called polymorphonuclear phagocytes, and these come from the bone marrow. They are myeloid cells. They develop under the influence of granulocyte CSF, right, colony stimulating factor. They are about 12 to 15 micrometers in diameter, and they have a lobulated nucleus. So these have these granules called specific granules and eosinophilic granules. The specific granules do not stain with H&E stain, and that gives them the name neutrophil, versus compared to eosinophils or basophils that do stain with this stain. So what are these granules? These granules have various enzymes: lysozyme, collagenase, elastase, whereas the eosinophilic granules have some microbicidal chemicals like defensins and cathelicidins, and also have enzymes that help in their function like myeloperoxidase. Lifespan is pretty short for these cells, a few hours to few days in the blood. If they enter the tissue, they are going to be active for 1 to 2 days and then they die. Let's talk about mononuclear. So polymorphonuclear had multiple lobules of the nucleus, and then mononuclear means the nucleus has a single, it doesn't have those lobulations as seen in PMNs, right? So mononuclear cells are also phagocytes, and there are monocyte-derived macrophages that go into the tissues. So they come from the bone marrow, develop in the blood as monocytes. When they enter the tissue, they are called macrophages. Compared to the monocyte-derived macrophages that come from the bone marrow, there are also some macrophages that come from yolk sac. So basically, a fetal liver that gives rise to some of these monocytes. And so they are different in the form of, they take residence in particular tissues, they are specialized in certain ways, and that they have such a specialized function, they are known with different names in different tissues. Give me 1 second, sorry, I just got paged. So, sorry about that. So yeah, so the some of these specialized macrophages are reside in the tissues, come from the fetal liver, and get these special names such as microglial cells in brain, alveolar macrophages in lungs. They are called Kupffer cells in liver, myocardial macrophages in heart. There are some again sinusoidal macrophages in spleen, and so on and so forth. These are long-lived macrophages that live in those tissues. This is information about where the macrophages come from in each organ, their origin, as seen in mice. Now remember, anything that's animal study related is typically not tested for you guys. This is just for information. All right, talking about these mononuclear phagocytes, in adults, we know most of the macrophages that are coming from the bone marrow, right? There are special macrophages for our tissue resident. We are talking mostly about the bone marrow derived mononuclear phagocytes. They develop under the influence of MCSF. They are 10 to 15 micrometers in diameter, and they have this bean-shaped nucleus, right? And then there is finely granular cytoplasm. So you see the first two pictures showing that bean-shaped nucleus, light microscope or the electron microscope picture. And then the third picture here is of an activated macrophage with vacuoles and cytoplasmic organelles. What are those vacuoles? The macrophages contain lysosomes, these phagocytic vacuoles, and that are important for their function. They are in the blood till they get to the tissue. Once they migrate into the tissue, they mature into macrophages, as we talked about. But we want to talk about the monocytes, the ones in the blood, right? There are different types of monocytes, and they are defined by their surface markers. So kind of keeping in mind that as we move forward, we'll be talking about a lot of these surface markers, because that's how we identify these cells. So monocytes are typically identified by the CD11B marker. They also have CD86 and MHC class 2 on them. So CD86 is a B71, B72. Remember, those are the co-stimulator molecules. So monocytes have this, which is CD80 and 86, B71, B72, two different names. And so this CD86 is a marker, also a marker on monocytes. And then, of course, they have MHC class 2, which is what they use for presentation of antigens. So kind of keeping these in mind, these are the cells that are identified by these cell markers, but they also have some cell markers that help us differentiate in various subcategories of monocytes. The classical monocytes that we talked about, those are CD14 strongly positive, CD16 negative, CCR2 positive. These produce inflammatory mediators and are recruited to the site of injury and infection. There they move out of the circulation, go to the tissue, and that's when they turn into macrophages. But then there are some non-classical monocytes. They are also mildly positive for CD14, strongly positive for CD16, and they are CX3CR1 positive. What are these CCR2 and CX3CR1? Those are the chemokine receptors. So the chemokine receptors tell them where to go, which chemokine to follow, right? So the non-classical monocytes crawl along the endothelial surfaces. What do they do there? They are kind of in a patrolling job where they are just looking out to and contribute, and wherever there is a need, they'll help in repair of the tissue after injury. Now, apart from these classical and non-classical, there is a third subset, which is called intermediate subset, because it's a combination of both of the monocytes. They are CD14 strongly positive, but they are also positive for CD16. In general, they are more inflammatory, so more like classical monocytes in their function than the non-classical. All right. So let's say the monocytes get into the tissue and they become macrophages. So macrophages, when they come across certain antigens, they will go ahead and get activated. There are two ways that they can get activated: the classical activation or alternative activation. Classical activation occurs in presence of cytokines that come from different cells like T cells, and they help in making macrophages more efficient killers, compared to alternative activation, where they are more helpful in tissue remodeling and repair. So rather than inflammation, they are the ones that are going to help in tissue repair. Now remember, macrophages are going to eat anything that they come across, to kind of, because these are sentinel cells in the tissue, and they are just kind of engulfing things that they come across. So why do they not just eat away host cells? So it's important to remember that there is a "don't eat me" signal that's present on the host cells that prevent those host cells from being eaten up by the macrophages. So this inhibitor receptor on host cells is called SIRP alpha, SIRP alpha, and it on the macrophages that recognizes CD47. So the CD47 is that "don't eat me" signal on the host cells, and the macrophages recognize that so that they do not eat those host cells. So what do these macrophages do? We know that they function in a couple of different ways. Macrophages are activated by microbial products like, say, for example, LPS, or they can be activated by certain cytokines such as interferon gamma. Once they are activated, so say here are all these signals that are trying to activate this macrophage. So either it could be on the microbial antigen that binds to pattern recognition receptors, or a cytokine that can attach to the cytokine receptor. There could be fragments attached to or tagged by complement, opsonized by complement fragments. So there are complement receptors on their surface. Also there, or immunoglobulin opsonized by antibodies. So there are various signals that can help in activation of macrophages. Once activated, they will release cytokines. Cytokines help in inflammation and increase the activation of the adaptive immune system. They also have some enzyme complexes that help them in their phagocytosis and killing function. So just like neutrophils, they also have phagocyte oxidase, in short called PHOX, so PHOX, PHOX, or it has this inos, or that helps in producing nitric oxide, and that also helps in killing the microbes. They can also help in tissue homeostasis and resolution of inflammation, as well as tissue repair, as we just talked about. So this table just gives you a comparison between the two types of phagocytes, neutrophils and macrophages. Remember, neutrophils are our foot soldiers. They get activated, they have a short lifespan, go to the front line to fight those microbes, and they get killed themselves. They are the first responders when they get the signal that they are needed, right? So they are pretty short-lived and quickly go ahead and go to the site of infection, engulf or phagocytose the microbes, and then they use the reactive oxygen species to kill the microbes. Compared to that, macrophages are long-lived in the tissue. They are sentinel cells, stay there for a long time, and then they are slow in responding to those microbes. Once there is a microbe that's engulfed, they will use their inos system more frequently to kill those microbes, compared to the PHOX system, which is more prominently used by neutrophils. The neutrophils can help in forming this net, or a neutrophil extracellular trap, where they extrude their nuclear contents onto the microbes. That also helps in killing the microbes, but they can also damage the host tissue with that. Macrophages do not do that. Macrophages are more prominent in producing cytokines that help in activating the adaptive immune system. They help in their function in general, in their phagocytic function. So they have a few differences that is important. Again, macrophages release help in activation of caspase-1 that produces fever. Neutrophils do not do so. Moving on from macrophages, so we talked about two types of phagocytes. Now we talk about mast cells. Mast cells are also produced from bone marrow. They develop under the effect of or influence of stem cell factor. Important to remember, anything in red in my presentations is for you to memorize. And that's basically for mast cells, the cytokine that helps in their development is stem cell factor, also known as c-KIT ligand. So what are mast cells? Mast cells have these granules in them that have various contents such as histamine and tryptase. They have receptors for IgE. So when those IgE antibodies on their surface get bound by various antigens, they can degranulate and release these contents. So the cytoplasm has these pre-found membrane-bound granules that contain various proteoglycans. And so usually these cells are present more in the tissue rather than in the circulation. They are close to the neurovascular bundle. That's where they reside. Compared to the mast cells, the basophils are very similar cells, right? These are produced in the bone marrow. They also contain granules that bind basic dyes, so that gives them the stain compared to neutrophils or eosinophils. And they also have mediators similar to mast cells, but mast cells are more prominent in the tissues and basophils are more in the circulation. They also express those IgE antibodies on their surface. When got bound, they can degranulate. Eosinophils are another cell from bone marrow. Myeloid progenitors they develop under the influence of GM-CSF as well. IL-3 and IL-5 contribute for sure. And then cytoplasmic granules contain basic proteins, so they bind the acidic dyes, and so they stain red. They are present in the tissues in mucosal linings. All right, the next one is dendritic cells. Dendritic cells, again, most of them come from the bone marrow. Stem cell that gives rise to either the common dendritic cell precursor, or they can come from the lymphoid progenitor or monocyte precursor cell. So they can kind of develop from different places. So the typical one comes from the dendritic cell precursor, and they form the classical dendritic cells, which are two different types. There are also plasmacytoid dendritic cells that come from the common lymphoid progenitor, and they're called plasmacytoid dendritic cells. And then there are some called monocyte-derived dendritic cells. These are all coming from the bone marrow. There are some that develop during the fetal life, and they can go and reside in the tissues, such as they can go into the skin and reside as Langerhans cells. Langerhans cells are known for the presence of granules called Burbeck granules. So dendritic cells, again, coming from bone marrow, and then they develop under the influence of FLT3 ligand. They, as the name suggests, have dendrites or long membranous projections. They also have different functions based on the type. So classical or conventional dendritic cells, they are antigen presenting cells. These are classical antigen or professional antigen presenting cells. Where are they? They are present in skin and mucosa and organ parenchyma, and they are waiting to see if they come across an antigen that they can present to the T cells. So they take that information from the tissue of what kind of antigen and where is it coming from and where is it located, travel into the lymphatics to the lymph nodes, and then they present that antigen to the T cells. Um, Compared to the classical or conventional dendritic cells, the plasmacytoid dendritic cells are important for response to viral infection. They are the factories that produce interferons, type 1 interferons. All right. So this table again, the pictures give you information about what dendritic cells look like with those dendrite out projections that you see. Um, Pretty prominent up here. If you can say prominent up here, and this picture here shows the Langerhans cells and the Burbeck's granules. All right, so dendritic cells, this table shows you the comparison between different kinds, and you can see some of the surface markers that are important for these cells, how we recognize these CD11C or CD11B, and then some other ones that are mentioned. Langerhans cells also have Langerine, CD207. That's important to remember. All right, so you have these are antigen presenting cells, so they have various pattern recognition receptors on their surface. That's how they get the information that's important for them to present those antigens to the T cells. They do have these transcription factors that are important for their maturation and function, and then the cytokines that we produce are different based on the subtype. As you see, most of them take part in innate immune response. The classical dendritic cells type 1 are important for something called cross presentation, which means we'll talk about this presentation. We did touch upon that during our discussion in SOMPIRAC, where we talked about how the antigen presentation works for CD4 and CD8 T cells, but here the antigens that are typically presented to CD4 are instead presented to CD8, so that's called cross presentation. So they help in adaptive immunity building, except for that most of the other ones have innate immune response. So these are important antigen presenting cells. What are the other professional antigen presenting cells? Those are macrophages and activated B cells. So 3 professional antigen presenting cells: dendritic cells, macrophages, and activated B cells. Other than these 3, there are not the professional, these are the 3 professional ones, but there are other cells that present antigens, like all nucleated cells can present antigens using MHC class 1 for CD8 response. Also, there are follicular dendritic cells, and follicular dendritic cells have nothing to do with the dendritic cells that we just talked about in terms of where they come from. So they are not from the bone marrow. These are found in the lymphoid follicles, and they present antigens to B cells. Talking about lymphocytes, lymphocytes are produced in the bone marrow from lymphoid progenitors, and we know that there is a very large number of cells. When we usually talk about lymphocyte counts, as we saw in the first slide, we were talking about blood circulation. Now we are looking at a huge number of cells present in the total overall body, and 65% of those lymphocytes are in lymphoid organs, right? So we see 2% of the lymphocytes in the blood. So all those lymphocytes the numbers that we looked at in the blood were only 2% of the total lymphocytes in the body. And of course, they are in other organs as well. So these lymphocytes are clonally distributed. They have diverse and specific antigen receptors. There are, of course, 2 types of lymphocytes, B and T cells, coming from the bone marrow. The B cells have subtypes: follicular B cells, marginal B cells, and B1 B cells. So follicular B cells are the ones that we typically talk about as B cells, but there are these other subtypes that we'll touch upon. T cells are again different ones: alpha, beta, gamma, delta, NK T cells, And typically the ones we call T cells are the alpha beta T cells that we talk about. Those are either the helper T cells or the CD4 T cells or the CD8 T cells or the Tregs. And then gamma delta and NK T cells are more part of the, have an more innate function than adaptive. All right, so looking at the various subtypes, the important ones are the alpha beta T lymphocytes. These could be CD4 positive or CD8 positive. CD4 positive help in action of macrophages or B cells. And of course, their markers are CD3 and CD4, right? CD8 have CD3 positive and CD8 positive. They also have alpha beta T cell receptor on their surface, and they interact with class 1 MHC. Compared to CD4 that interacts with class 2 MHC on the APCs. And the CD8s help in killing the intracellular microbes or tumor cells. Tregs are another type of subtype of T cells. These are also alpha beta. They are CD4 positive, CD3 positive. They are also CD25 positive and FOXP3 positive. So important markers to remember: CD25, FOXP3 define the Tregs. NK T cells have the surface markers for NK cells and T cells. So they have CD3 on their surface. They also have CD16 and CD56 on their surface, which are the markers of NK cells. They do have alpha beta TCR on their surface, but they have limited specificity compared to the specificity seen with CD4 or CD8 T cells. The other type of T cells are gamma delta. Instead of alpha beta, their TCR has gamma delta, and they are CD3 positive, but CD4 and CD8 can be variable. There are cells called MAI T cells or mucosa-associated invariant T cells. These are also alpha beta T cells, but again, like NK T cells, they have limited specificity, and these are mostly CD8 positive, CD3 positive cells. B cells, subtypes: follicular B cells, marginal zone B cells, and B1 B cells. Follicular B cells are our typical B cells. They have CD19 that defines the B cells. They are important for all of these B cells are important for antibody function and humoral immunity. The follicular B cells have diverse specificities. Compared to that, the marginal zone B cells have limited specificities, just as are B1 B cells. And so on their surface, you will find IgM. For marginal zone B cells, they have CD27. B1 B cells, they also have other markers mentioned here. Again, just kind of keeping in mind, follicular B cells are the important ones that are the classical B cells, but the marginal zone B cells and B1 B cells have limited specificity for restricted number of molecules. Where do these lymphocytes develop? They develop or come from the bone marrow, and they mature in organs such as thymus, or they go into the periphery, and the cells destined to become B cells mature in the spleen. So important to remember, primary lymphoid organs are the ones that help in generation of lymphocytes, and they provide those cytokines and antigens that are self-antigens that are required for that production of lymphocytes as well as development or maturation, compared to the secondary lymphoid organs like lymph nodes, spleen, mucosa, or cutaneous associated lymphoid tissues. That's where the mature lymphocytes go, and they come across the antigen that help in their activation. So production and maturation occurs in the primary organs, and then for T cells for sure, and for B cells, some of it in the maturation occurs in spleen, and then the secondary lymphoid organs is where the lymphocytes get activated. Talking about various stages of the lymphocytes. So the stage that they develop in and finally mature are called naive cells. When they come across their antigen, they can get activated. Those are called activated lymphocytes. When they differentiate into something called effector lymphocytes. And then once they undergo their function, the ones that are left behind and keep the memory of that infection are called memory lymphocytes. So keeping those four stages of a life cycle of a lymphocyte for both B and T cells, but there are several differences, and those are important differences to remember. The lymphocytes, when you find them in certain places, they would be in different stage of their life. For example, the naive T cells preferentially recirculate to secondary lymphoid organs, whereas the activated or effector cells go to the tissues or inflamed tissues, and then the memory T cells can go to the inflamed tissue and mucosal tissues as well. Mainly looking at the surface markers. These are really important to memorize, and they have function that you will learn about and that will help you memorize this information as well. So the surface markers that are important or define naive T cells are CD62L, CD45RA, and CCR7. Those three are the markers that are used to define naive T cells. Of course, they also have some IL-7 receptor as well, but that can be high even in the memory cells. Compared to the naive cells, the activated T cells have IL-2R or IL-2 receptor alpha, also known as CD25 on them. That is a good marker for activated T cells. They can also have CD45RA RO on them, as well as adhesion molecules, integrin CD44 that's high in them. Memory T cells, on the other hand, have high IL-7 receptor, the CD44, as well as CD45RO. So CD45RO is also used as a marker for activated and memory T cells. So again, important to remember and memorize. Markers for naive T cells are CD62L, CD45RA, and CCR7. B lymphocytes. So again, the various stages of life of a B cell, those are naive, activated, and memory cells. So the naive B cells have IgM and IgD on their surface. Activated B cells may initially have IgM on their surface, but then they can class switch to having IgG, A, or E on their surface. Same for the memory B cells. The affinity improves once they get activated and they undergo hypermutation. And surface markers that are important, CD27, again,